Dear friends, good morning to all. Today we can see active site and mechanism of enzyme action. Active site. Definition of active site. The active site of enzyme is the reagent that binds to substrate and contains the residues that directly participates in the making and breaking of bonds. The residues are called as catalytic group or active site. General characteristics of active site. Although enzymes differ widely in structure, specificity, mode of catalysis, the active site takes up a relatively small part of total volume of enzyme. The diagram represents the active site of enzyme. And the enzyme which is outer surface having the active site, this is the substrate binding site of enzyme molecule. Substrate which reacts with active site of enzyme combine together to form the enzyme substrate complex. Most of the amino acid residues in the enzyme are not in a contact with substrate. The active site is a 3D entity. 3D entity means three-dimensional structure. It is formed by different parts of the linear amino acid sequences. Residues far apart in the linear sequences may interact more strongly than adjacent residues in the amino acid sequences. Substrate is bound to enzymes by multiple weak interactions, van der Waals force and hydrogen bonds. Active site is a cleft or cervix. In all enzymes, the substrate molecules are bound to cleft or cervix. The non-polar characteristics of cleft enhances the binding of substrate molecule. Then the specificity of binding. It depends on the precisely defined arrangement of atoms in active site to fit into the site. A substrate must have matching shapes of the active site of the many enzymes are markedly modified by the binding of substrate. Functional groups which are present in active site. Carboxylic group of dicarboxylic acid, terminal carboxylic group of polypeptide, amino groups of lysine, guanidine group of arginine, indole group of tryptophan, imidazole group of histidine, hydroxyl group of serine and threonine, SH group of methionine, phenolic group of tyrosine. These are the functional group of active sites. Next, move on to mechanism of enzyme action. Here we can discuss two theories. One is Emil Fischer lock and key theory. Another one is Kausland induced fit theory. First, we will see about lock and key theory. In 1890, the scientist Emil Fischer suggested that the enzyme specificity impelled the presence of complementary structural features between enzyme and substrate. As substrate might fit into its complementary site on the enzymes as a key fit into locker. Active site of enzyme is pre-shaped and also rigid form. This is the diagrammatic representation of lock and key mechanism. Here enzymes which have the active site, substrate also having the binding site, which are complementary to each other. Active site lock and key mechanism. Enzymes reacts with substrate as a key fitted into lock. Negative features of lock and key hypothesis. Rigidity of active site failed to explain the reversible reactions. Next one, induced fit theory. Kausland modified Fischer's lock and key mechanism in 1963 in the form of induced fit mechanism. The essential features of this model is the flexibility of reagent of active site. Unlike the lock and key mechanism, according to this theory, the active site has shape complementary to that the substrate only after substrate is bound. Cauchy induced fit theory, the enzymes does not retain its original shape and structure complementary to that of substrate, but contact to the substrate induces some conformational or geometrical changes in the active site of enzyme molecule. Consequently, the enzyme molecule is made to fit complementary the configuration and active center of substrate. The process is dynamic reorganization is called as induced fit. Here the diagrams represent the induced fit theory. Enzymes active site differ from substrate binding site. Near to the substrate, then only the conformational changes takes place in active site of enzyme. Then enzyme substrate complex formed. Thank you.